concentrate loci for plant height heading it into specific process of the rice and comparison across the rice genus. Please welcome.
Good afternoon, everybody. It's my pleasure to give my recent work here, and also thanks Dr. Epicha to give me this opportunity to present it. Okay, so actually from this 3 days or 4 days conference that we know Dr. Win brought out a very important dream that 9 billion to solve the 9 billion people questions, right? So during his question that we try to breed high yield or drought tolerance or the other insect resistance genes to reach the goal. So yield is the major breeding goal in feed more people in the near future. However, I think grain quality is still a major breeding goal in different countries or in different areas. Like we know that we had the glutinous rice last night in the banquet. Okay, so glutinous rice to me it's very odd that why it can be a major food in the regular dinner, but in the northern Thailand and the northern eastern Thailand, people eat the glutinous rice every day. So in this situation, and under this consumer requirement, the major university nearby here start a program try to breed new varieties with the glutinous. So they introduced a non-functional wax gene to 3 elite cultivars. I forgot the name. It's not easy to remember. I tried to remember it. Yeah, the Patantani and the Chatnet 1 and the KTML 105. So to these 3 elite cultivars, they breed 3 elite cultivars now. So it's major 2 and major 4 and the major 6, especially major 2 won the award this year. So it means that different countries or different areas has their preference for their grain quality. In Taiwan, actually our breeding goal has been changed to grain quality. One of the reasons that the production in Taiwan is really high, actually maybe we can call overproduced. So the grain quality breeding program has been started maybe since 1970. So in this situation, we have to find efficient way to evaluate the phenotype, because it's not easy just ask people to eat and evaluate. So in this situation, we try to find the indirect index for our grain quality selection. In that situation, that we think palatability and the viscosity are very important for the cooking and eating quality. And we know that many genes are involved in these two characters. So we start to do this program. Yeah, and we hope that our study can be used in breeding program. And in addition, we try to find more genes that can regulate this grain quality and for the basic research. And this work is collaborate from National Taiwan University and the Taiwan Agricultural Research Institute. Dr. Wu is a breeder in our country. Okay. So for the rice grain quality, that we always consider about 3 aspects. One is the milling quality. Is counted on the head rice after the milling processing. The other one is grain appearance. We always prefer the non-opaque or non-chocolate grain. So these 2 are very important. But for the consumer preference, I think eating and cooking qualities are very important. So we always evaluate by generalization temperature or gel consistency or amylose content to try to use this kind of index to infer the eating and cooking qualities. But here we use the palatability and the viscosity to evaluate the eating quality. From the rice grain that we can know, a majority of the rice grain are composed of starch is over 90%. The other part are protein and lipids too. And the starch in the rice grain or the starch in the nature starch that composed two different polymers. One is amylose, amylose is alpha 1 for linkage of the polyglycan, and the other is alpha 1.6 linkage of the for amylopectins. In this indica rice, there are more amylose content, over 25% amylose content. So when we eat indica rice, it's more fluffy. It is not so sticky. And for japonica rice, like the amylose content is about 16 to 20%. For the wax gene, there is only maybe less than 2% amylose content. For the other part are amylopectins, but it's easy to measure the amylose contents. So sometimes we infer that the structure is by amylose content. Okay, so these are just a very brief review how the starch can be formed in a rice grain. So after the photosynthesis, the glucose are transported into amyloplast. After that, GBS1 protein are in charge to synthesize amylose. And for the other part is how to synthesize the amylopectin. Actually, there are lots of different enzymes are involved, such as starch synthesis and starch branching enzymes and starch debranching enzymes. All these enzymes together, right, from these different kind starch structures. So. And the last part is how to get the crystallization. Yeah. So it means that all the processing here all involve the grain quality. But we are still curious about which genes already account for grain quality, especially eating and cooking qualities, like these genes exist in the natural variant. And are there any other genes beyond starch synthesis also control the grain quality? Is our question to brought up. So maybe about 20 years ago, Dr. Wu screened 24 indica rice and 30 japonica rice to test how their eating and cooking qualities by physiochemistry studies. So among these 54 varieties, we found out that the japonica Taiwan 72 and the Taizhongxian indica Taizhongxian 17 has quite different at the amylose content. So Taiwan 72 is a japonica, has lower amylose content. Taizhongxian is indica, has a lot amylose content. Beyond the amylose content, actually we also see a lot quite different variation between these two varieties, such as a setback or peak viscosity or the others. So we started this program to use these two varieties as mapping parents. However, not only use the Taiwan 72, we use a new variety in Taiwan 78. Taiwan 78 is a new variety with a joint embryo that you can see from this photo. It's derived from the point mutation from the Taiwan 72 induced by MMU. So in this study, we use a Taiwan 78 instead of Taiwan 72 for our mapping population. So we started this course at 2005, the first season, and we are so lucky we planned 2 seasons of rice in Taiwan. So like in the second crop season of 2010, and we have recombinant invariance. We use only 190 recombinant invariance for the genotyping, and we also did the phenotyping for these 2 different crop seasons. So then the phenotyping is evaluated by the palatability and RVA characteristics. So this in the field in Jiayi. Yeah, so these are 2 machines that we use. This is a test analyzer, like it can give evaluate the palatability score. The higher, the better. The other one is rapid visco analyzer, RVA. actually, this one has been used by breeders to evaluate the cooking and eating quality very well. So these are different characters that we used. First is a pasting temperature that stock granules start to swollen. And then the peak is the time and the peak time and the peak viscosity and the hot then show it by RVM machine. Then we test the hot paste viscosity and the temperature is cooled down. Then we got the cold pastability and the breakdown viscosity and also the setback viscosity. All these 8 properties that we use to analyze the QTL. So from the histogram here, that we can see a lot of transgressive segregation in these 2 different seasons, and also among these different 8 characters. In addition, that we also see the bimodal distribution at several traits. So it means that they are these traits are polygenic inheritance, and maybe one major gene control these bimodal distribution traits, and the other one that we see the different distribution among these 2 crop seasons. So environment definitely influenced this trait expression. So we did the correlation analysis here for these two crop seasons. Some traits are really highly correlated, and some maybe not. So for the highly correlated traits, we expect there are some conserved QTL controlling. Yeah. So we start for the recombinant line for the QTL mapping. So we use 133 PCR markers. So some of them are some markers or indel markers, and we also include 6 starch gene tag the markers here. So a total linkage map is 1500, and the average between 2 markers 11.4 centimorgan. Because this cross between indica and japonica, definitely you will see a lot of segregation distortion in the recombinant population. Yeah, so we use a composite interval mapping to reveal the to uncover the QTLs among these 8 traits. So we can see there. So there's a gene clusters along the chromosome 6, and also the other gene clusters around chromosome 7 and 10 here. So it means that there may be at least a pleiotropic effect. One gene can affect so many different characters. The other things that you will also see the environment effect that at least QTL only detect in 2010 second crop season, and this QTL may be also only specific for that trait. So we make a summarized here. Actually, we also put a wax gene encoded to GBSS1 enzymes, which is important for amylose content, amylose synthesis. We find this gene has the largest effect. Yeah, from about 30 to almost to up to 80%. So wax gene actually contribute a lot to these different characters, and it's also very conserved among 2 crop seasons, and also among the different characters. And the other QTLs maybe is a season dependent and also different characters specific. So in these 2 crop seasons, actually there are 10 pairs are conserved, but the others are not. So a total we found 24. Yeah, and here that you find that the Taiwan even is an indica, but it still can contribute good alleles. But most of the part that is Taiwan 78 provide good alleles for breeding. Yeah. Up to now, we only find maybe more, but in that a couple months ago, we only found 3 papers also discuss this study. So from these 3 papers that we found the chromosome 6 the QTL on chromosome 6 are really stable, not only in our two different environments, also stable in different studies, and also the QTL on chromosome 7 too. Yeah, and we think about that maybe gene effect, it can be not detected by QTL. So we use a gene as a marker and to do the single point analysis. Actually, WEXC gene contribute a lot. So we under this WEXC gene that we see the distribution actually can be divided to two different groups. And in that situation that we use a WEXC gene like a cofactor to see the gene interaction. Actually, these gene interaction are haplotype independent. So like the pollinase 1, the SP1 is only interact in the indica type, but not for japonica type. So this provide an important thing that if you want to do a japonica breeding or indica grain quality breeding, then we have considered different genes. So and after QTL mapping, we want to know which genes are involved to these characters. So we do the gene expression. Let the genes locate and also segregate in this region. So to see if their gene expression are different. Actually, from here, like the SPE4 pollinase, ISA2, and the GBS1 show quite significant difference between these two parents. But not for SS2A. And this one that we did not detect by QTL, but it also show difference. So it means that maybe the power is not enough to detect that one. So from the starch synthesis here, right, actually GBS1 encode gene that show has a lot of effect because it amylose. For the others that involve to starch synthesis, branching enzyme, debranching enzymes, here also show effect to affect the amylopectin composition. So from here, right, we can make a conclusion that actually grain quality is still our breeding goal now. And in this study that we find the 10 pair stable express QTL environment effect definitely is here. And GBS1 has a largest effect. And the gene interaction is haplotype dependent. And we find that there are several genes involved in starch synthesis also control or influence that the grain quality. So it means that if we want to improve the grain quality of the elite cultivars, actually we can base it by the genes that involved in starch synthesis. However, we need more genes like the QTLs on chromosome 7 and on chromosome 10 show clusters for also pleiotropic effect to different characters. However, none of them are corresponding to any genes involved to starch synthesis. So it means that more genes beyond the starch analysis. And Su Zhen in Taiwan Agricultural Research Institute helps to manage the field work. And Fujin and Caroline help us to mine the polymorphic genes. Yeah. Regarding to like the starch synthesis, yeah, and the findings from Taiwan National Science Council. Okay, thank you.
We have time to entertain just one question. One question? No. Dr. Lin, this is very easy. I confuse the word palatability. So how can you define it here? Palatability.
Palatability, it actually is we use taste analyzer that the machine is invented by Japan. So it's like can mimic the texture of the rice grain. So it means that you use like a light transmit to the grain, cook the grain, and the water retained on the grain will reflect the light. So they use this method to measure how many water still retained after cook on the rice. So it means the water content in the cooked rice can reflect the texture. Yeah, so I think it is a simple.
I'd like to know that the one that machine like will be the one that we like. The one that your taste analyzer like will be the one you like or the one I like. Maybe not the same one. I may like something else and the machine likes something else.
Really? So look at the models here. Maybe I used the wrong because the machines belong to the breeder. I should ask him. Sorry. The machines belong to the breeder. I should ask him give me the photo. So when I find the photo, I just get it from.
Just kidding.
Sorry.
We all have delicacy about the taste, right? So when machines say this is the most palatable, means that we would like. Okay, thank you very much.
Okay, thank you.
Have a big hand to Dr. Lim.
Thank you.